From 2001 to 2011, the Zabolotny Institute of Microbiology and Virology of the National Academy of Sciences of Ukraine carried out complex researches of microbial ecosystems of Western Antarctica. Much of the researches were carried out by employees of Department of Extremophilic Microorganisms Biology. During 10 years, we have been studying the ecosystem in general and microbial ecosystem particularly in the area of inner and outer island archipelago, as well as adjacent to the Ukrainian Antarctic station Academic Vernadsky of Antarctic Peninsula. The complex researches of Antarctic microbial ecosystems included three serial stages. The first, it is the fundamental researches. The second is the bioprospecting, and the third is an approbation of technologically perspective microbial strains. The main goal of fundamental researches was to study the physiological and ecological trophic groups of microorganisms, as well as their quantitative and qualitative calculation. The task of bioprospecting was to isolate the produces of biologically active substances and also isolate perspective strains to develop new environment protection biotechnologies. Finally, the approbation, it means the testing of collection extremophilic microorganisms in the environment protection biotechnologies and technologies of biologically active substance synthesis. Thanks to the invaluable assistance of Chilean navies, we were able to explore the islands of the external and internal archipelagos, snow-white faraway places. At first sight, the Antarctic Islands and Antarctic Peninsula is a lifeless space, only blinding snow and ice. There were no signs of life. However, at more detailed study of the islands, we found huge fields of creatinosis formed by the algae bacterial mates. The 10th of December 2010. We are on the Antarctic Peninsula in the point called Brown Bluff. It is formed from volcanic rocks. The rocky cliffs amaze us with powerful covers of yellow crustous lichens. The continuous thick layers of lichens cover the hectares of vertical cliffs. The height of tough cliffs is more than 100 meters. Above the cliffs there is a glacier, the water source that is necessary for the development of biofilms on the rocks. At temperature above zero, melted glacier water washes the surface of cliffs by continuous flow, and it leads to intensive growth of yellow crusted lichens. Intensive falling of protruding rocks with lichens layers leads to the fact that the stones under the pressure of life, or rather the biomass of lichen, separate from the cliff and fall down into its base. Antarctic volcanic island deception is represented by dead landscape. There are naked cliffs covered with the volcanic ash with no any visible signs of life. However, some areas are inhabited by the squab birds. Their constant presence determines the biological attack of volcanic landscapes. On rocky juts, in places of squab rest, accumulate the excrements. Excrements, the excellent organic substrate for growth of microorganisms, lichens, mosses, and finally for colonization of lifeless volcanic landscapes. On the top of the rock take place the modern process of colonization. The pioneers of colonization are microorganisms, microscopic algae, maybe some other algae, the crusted lichens, and branched lichens in combination with yellow crusted lichens. Listed factors provide the necessary condition for growth of highly organized plants, mosses. The lower the slope, the more mosses layer of biofouling, and the more developed concentration of organic matter, and apparently we can say that from the top to down in such sloping landscapes occurs a carbon gradient, including the closed carbon cycle. In general, our well-known biogeographical polygon on the island Galindez, that is subpolar thermostated oasis, where the life is literally rages and all possible variety of Antarctic ecosystems are represented. The same patterns were observed for the biogeographical polygon and the small Tooth Cape on the island Deception. We gave the several examples of the intensive ecosystem development in the harsh Antarctic conditions. However, the most extreme, amazing ecosystem we have found in the Paradise Bay as the output of concentrated copper geological structure on the rocky cliff. This copper structure is washed by water stream, and this water saturated with the copper compounds runs on a rocky shelf. The most amazing was that the copper saturated stream filtered through a layer of living lichens and moss on the rock bottom. Because of little camera resolution, it was impossible to obtain a clear image of the extreme ecosystem, and the large height of vertical cliffs, which is about 100 meters, excludes the possibility to get the copper formation. But observed ecosystem probably is one of the most extreme in Antarctica. We carried out the fundamental researches of the structure and function of microbial ecosystems in the area size of 30 by 60 kilometers. This zone consists of dozens of islands and coast of the Antarctic Peninsula adjacent to the academic Vernadsky station. One of the main characteristics of ecosystems is so-called homeostasis. It is the ecosystem ability to keep and support its functioning under the pressure of extreme factors. As such factors, we choose 6 toxic metals: chromium, nickel, copper, cobalt, cadmium, mercury, in concentration range from 100 to 5000 ppm. The maximum permissible concentration of these metals for microorganisms will be in quantitative parameters of homeostasis of microbial ecosystem in Western Antarctica. In this context, it was necessary to answer 2 main questions. The first, how widely metal-resistant microorganisms in the area of size of 30 by 60 kilometers are spreaded? And the second question, what is the upper concentration limit of microbial resistance to the 6 most toxic metals in this zone? After a decade of researches, we have obtained the following data. The first, metal-resistant microorganisms were found out in all 19 geographical objects in the area in size of 30 by 60 kilometers. The second, Antarctic microorganisms can grow at bactericidal concentration of 6 toxic metals. And the third, microorganisms from Western Antarctica are characterized by polyresistance, that is simultaneous resistance to several specified metals. This implies that the metal-resistant bacteria are widespread in the investigated area, and metal resistance is inherent to most microbial ecosystems. On this diagram are shown the generalized data of metal-resistant microorganism distribution, typical habitats of ecosystem in the area of internal island shelf, biotopes or ecosystems, other soils, lichens, mosses, lake sludge and humus. The blue arrows show the number of samples in biotope resistant to mercury, chromium, cadmium, which is in the range from 30 to the 85% of all tested samples. It should be noted it was studied about 260 samples, and representative concentration range of metals was bactericidal for the vast majority of usual hemorganotrophic microorganisms. For mercury it was 50 ppm or 50 milligrams per liter, and for cadmium and chromium it was 500 ppm. To describe the quantitative characteristic of microbial metal resistance, we used two parameters. The first is the total amount of microorganisms or colony-forming units cultivated at the given metal concentration in sample. The second is the occurrence coefficient of microorganism resistant to given metal concentration. The coefficient was calculated as the percentage ratio of metal-resistant microorganisms to the total number of microorganisms cultivated in the absence of metals. On 12 geographic objects it was shown that the total number of metal-resistant microorganisms was very high, taking into account that the metal concentration were bactericidal. For example, in samples selected from the island Uruguay, the number of microorganisms resistant to mercury in concentration of 50 ppm was the tens of thousands of cells per gram of sample. The same colony-forming units number was obtained at 500 ppm of chromium and cadmium. The very high values of occurrence coefficient of metal-resistant microorganisms were determined towards chromium-6 at concentration of 500 ppm for island Yelur, it is 5%, and for Cape Rasmussen, it is 2% in the case when the concentration of chromium-6 was even 1000 ppm. Pilot table shows the generalized data which indicate the high values of both resistant parameters of microorganisms to chromium, mercury and cadmium. In a number of island ecosystems of Argentine archipelago, microorganisms show a high level of metal resistance. For example, increasing of chromium and cadmium concentration from 20 to 500 ppm do not affect the number of resistant microorganisms. The total amount of viable cells is constant. On the right, on figure, it is shown the area of size of 30 by 60 kilometers, in the middle of which is situated the Argentine archipelago and the island Galindez, where the Ukrainian Antarctic station Academic Vernadsky is located. On this island is situated a biogeographical polygon, which were founded in 2002. On biogeographical polygon, we identified a local polygon. It is a rocky cliff with height from 10 to 12 meters and called Scaladrome 2. Thanks to the captain of Chilean warship Viel, Colonel Artura, pilots commander Army Major Romana, and brave pilots, in 2008 we were able to make a panoramic video shooting of the biogeographical polygon on the island Galindez. The main object of our researches is the biogeographical polygon on which we have been carried out complex researches for 10 years. The polygon is a rocky cape with a maximum height of 58 meters. At the top of the rocks, coupled with the relic glacier, then pass into large sloping moss fields with soils, grass and birds' nets, and then pass into the rocky precipices or cliffs. The height of cliffs varies from 10 to 20 meters. At the bottom of the polygon, a lake with large depositions is situated. Microclimatic features of polygon determine the maximum biodiversity of all ecosystems. As a result of the low height of polygon, which is 58 meters, and at positive air temperature, an intensive snow melting occurs on the glacier. Therefore, all ecosystems below the glacier obtain the constant water feed. The presence of algae bacterial mates or creatinoses, the huge moss fields, indicates about the favorable conditions necessary for the development of Antarctic ecosystem on polygon. The photosynthetics, microscopic algae, rapidly proliferate on the glacier surface. After rotting off, the algae biomass is partially fermented by microorganisms,
forming the humus, and then ice, soil-like substrates. Much of carbon is converted into carbon dioxide and methane. As a result, the ice creosonosis can significantly increase the amount of greenhouse gases in Antarctica. During 10 years in creation of a top base of biogeographical polygon were involved employees of the company ECOM, President Evgeny Seredinin, Vice President Dmitry Mizin, geodesist Yuri Lednovsky and Vadim Kurenkov. Viktor Usenko, PhD of Geological Sciences, made the detailed geographic description of polygon, the choice of landscapes for positioning. PhD Geographic Sciences Vladimir Grishenko conducted the system researches of the glacier on biogeographic range and set the significance of glacier in the formation and development of polygon ecosystems. Lina Kobzar started the system work on developing the database with the characteristic of ecosystems. Anna Berezkina began to combine the biomarker thematic layers of ecosystem with landscape elements of biogeographical polygon. With the help of GPS and GIS methods, it was developed a topographical map and 3D stereometrical model of polygon. On these maps, the main landscape elements, 210 points of long-term monitoring and typical biotopes of ecosystems are represented. During 10 years, members of seasonal scientific expeditions carried out the system biological, geodetic researches and selected the samples for subsequent microbiological analysis of polygon ecosystems. On the biogeographical polygon, mainly developed all representative Antarctic ecosystems such as mosses, Deschamps Antarctica, crusted lichens and various other forms of lichens. This scheme shows integral biogeographical model of polygon representing the zonal distribution of ecosystems in vertical direction. We have proved that for representative ecosystem is characteristic the high concentration of hemorganotrophic microorganisms. In all ecosystems, the number of cells was in order millions and even 100 millions per gram of samples. For the first time on polygon, we have performed the complex researches of Antarctic ecosystems on the quantitative and qualitative calculation of ecological trophic groups of microorganisms. We have shown the wide biodiversity of microbial zoonosis in relation to such factors as temperature, oxygen, and organic substances concentration. Important for the characterization of biogeochemical process of subpolar Antarctica is the ratio of psychrophilic and mesophilic microorganisms. We found that the ratio of psychrophiles and mesophils in ecosystem is almost equal. Consequently, the biogeochemical cycles of elements transformation in these ecosystems can be carried out by microorganisms in wide temperature range from 0 to 28 Celsius degree. In Antarctic biotopes or ecosystems, there are at least 7 genera of bacteria and 1 of yeast. We have selected the pure cultures of typical representative of the Antarctic microorganisms as Brevibacterium antarcticum and Exophiala nigra. On the islands of Inia archipelago and biogeographical polygons, the microbial zoonoses are diversified. It confirms the closed carbon cycle carried out by soil microorganisms. The number of microorganisms that belongs to the physiological groups of carbon cycle is very high, and in the range from 100,000 to tens millions cells per gram of sample. The same characteristic is for microbial nitrogen cycle. The cycle is closed and the number of microorganisms is in the range from tens thousand to millions cells per gram of sample. We study the homeostasis or resistance of Antarctic microbial ecosystems in extreme conditions using the factors such as ultraviolet radiation, the wide spectrum of toxic metals and antibiotics. Testing of physiological and ecological trophic groups of microorganisms allowed us to obtain quantitative parameters of microbial resistance to these factors. A 2004 geochemical analysis of soil from polygon and islands of Argentine archipelago showed that toxic metals are present in biotopes in trace amounts. The exception was copper, the concentration of which was several hundred of milligrams per kilogram of soil. Therefore, we initially assumed that the microbial resistance to metals will be minimal. However, later we established that the metal resistance of Antarctic soil microorganisms exceeds several times their local Clark concentration. For example, mercury and chromium concentration in soil was 0.2 and 2.3 milligrams per kilogram of soil, while soil microorganisms showed resistance to 500 milligrams per liter of mercury and even unbelievable 60,000 milligrams per liter of chromium-6. Arise the question, how in the most non-contaminated area of the globe can appear such microbial resistance to a wide range of metals? We can only assume that the metal resistance in Antarctic microorganisms appeared in the era of neotectonic activity in Antarctic, and this resistance is at the genetic level and is present in spite of the very low concentration content in ecosystems nowadays. We carried out a quantitative calculation of metal-resistant microorganisms in different polygon ecosystems in increased concentration gradient of toxic metals. On these photos it is shown that the increase of concentration of K2 cation up to 1000 ppm leads to decrease in the number of microorganisms in comparison with control only in one order, from 10 million to 1 million cells per gram of sample. Further, we found out that the copper-resistant microorganisms widely spread at the polygon. Points with microorganisms resistant to copper concentration of 100 ppm and higher marked on the map in red color. On the stereometric model of polygon is shown that the copper-resistant microorganisms distributed in all ecosystems, such as cliffs, crusted lichens, moss, grass, soil, sludge. Their number is in the range from 10,000 to 10 million cells per gram of sample. Then we determined the quantitative parameters of microbial resistance to such metals as mercury, cadmium and chromium. The number of resistant microorganisms to these metals was in the range from 1,000 to million cells per gram of sample. This stability manifested at high concentration of metals, 50 ppm of mercury 2 and 500 ppm of cadmium and chromium. From soil samples, Nadezhda Matveyeva and Anna Tashyrev isolated 10 strains of microorganisms resistant to extremely high concentrations of the 5 most toxic metals. Microorganisms grown in the presence of 500 milligrams per liter of mercury, 1000 cobalt, 2000 nickel, 2500 copper, and even at 60,000 milligrams per liter of chromium-6. On the basis of system researches, we have shown that in Antarctic subpolar oasis is widely spread microorganisms which are resistant to 6 most toxic metals at concentration on several orders exceeding their bactericidal concentrations. It implies that the ecosystem of subpolar oases is characterized by high level of homeostasis. Recall that homeostasis implies the ability of any ecosystem to maintain stability under the influence of extreme or stress factors. Phenomenon of multiplied resistance of chemoorganotrophs to most toxic metals in their concentration range from a few grams per liter up to tens of grams per liter still has not yet been described. Metal resistance of Antarctic microorganisms does not mean that they can involve the metals into the biogeochemical cycles of elements in Antarctica. It is necessary that microorganisms must be not only resistant to metals, but also will interact with these metals. We found out 3 types of interaction of Antarctic microorganisms with metals. The first is the reduction and formation of insoluble compounds. For example, the reduction of chromate anion to chromium hydroxide 3. The second is the reduction of iron 3 and further precipitation in form of sulfate iron 2. And finally, the reduction of copper 2 cation to insoluble cuprose 1 oxide. The following mechanism of interaction is metal mobilization. We found the copper hydroxide stabilization by microbial exometabolites. Finally, the third type of interaction is metal immobilization. We have shown the copper 2 and iron 3 accumulation in the growing colonies of microorganisms. Till now we have described microbial polyresistance of the first order. We mean that in each case of experiment in the culture medium there was only one metal, despite the fact that each of the microorganisms are resistant to several metals. According to our definition, polyresistance of the second order means the ability to grow while simultaneously the presence in the culture medium of several metals. As shown on the photo, Antarctic microorganisms are resistant to high concentration of 3 metals. The phenomenon of the second order microbial metal resistance is a reliable prerequisite for use of Antarctic microorganisms for purification of polymetallic industrial wastewater. Thus, polyresistance of the second order opens up broad prospect for developing innovative environment biotechnologies based on the Antarctic microorganisms. Microbial ecosystems are the ideal objects for modeling the changes in Antarctic ecosystems. Microbial ecosystems most quickly react on the changes in environmental factors. Microbial ecosystem of cliffs are maximally representative. During all over the year, the complex of extreme factors simultaneously and continuously influences on microorganisms of cliffs. These factors include UV radiation, toxic metals, low temperature, freezing, thawing, and many other extreme factors. Unequal distribution of organic matter on the cliffs leads to the phenomenon of antibiosis. Microbial antibiosis is manifested as synthesis of antibiotics and the formation of resistance to them. We will give the detailed description of Antarctic microbial ecosystem of cliffs on their resistance to a wide range of extreme factors. But to start the researches on the cliff, we should at least clean up the cliff and hang the rock outfit. Briefly we show how it has been happened.
geographical polygon, we have chosen as the main object of the investigation two cliffs and called them Scaladrome 1 and Scaladrome 2. At these cliffs take place the maximum development of all ecosystems. Vertical cliffs completely covered with a layer of moss biofouling. The maximum biodiversity is determined by two factors. The first is the presence of the glacier above cliffs, and the second, thermostatic mode of polygon. Under thermostatic mode, we mean the identical temperature at the given moment of the time, both as above cliff 58 meters height as above sea level. These two factors determine the constant intensive melting of snow and ice over the cliffs and therefore excessive ecosystem irrigation on cliffs. The photo shows that in the snowy winter Scaladrome 1 is completely covered with snow and cliffs of Scaladrome 2 is naked. Therefore, during all year the microbial cyanosis of Scaladrome 2 are exposed to a complex of extreme factors. Therefore, as the main object of research, we choose Scaladrome 2. On the cliff Scaladrome 2, except of the extensive areas of continuous biofouling, there are also naked zones of the rocks. On these rocks, at the present time, take place a process of active colonization of surface by microorganisms and lichens. Therefore, on the cliff Scaladrome 2, we have put two profiles, each of which passes as through the biofouling as through the naked zones.
This is the vertical polygon on which we worked on January 26 in 2008. This is profile 1. Here I went down from the top and further along the crack down to the base of the rock. On this segment was taken 5 points, more precisely 5 samples. To the right it is the rock section. As I remember, this example was taken from the top of this rock slab, completely covered by lichens. And then down to the point I took another sample, here on this rock covered by barbed lichens. And then at the base of the rock I took this black soil.
We have created a stereometric model of Scaladrome 2 and positioned 12 monitoring points on two profiles. That is long continuous observation. From samples taken from position point, we have isolated typical or representative microorganisms from the limiting dilutions. To obtain the quantitative parameters of resistance microbial ecosystems to the complex of extreme factors, we have investigated the polyresistance of representative strains to UV radiation, 5 toxic metals, and to the wide range of antibiotics. Now we take the samples on cliff, mainly on vertical rock cliffs. From these points we isolated the pigmented forms of microorganisms, the producers of biologically active substances. In particular, we isolated these producers of biologically active substances which possesses anti-ulcerose and anti-cancer properties. It has been shown by Professor Beregovaya. In our department of extremophilic microorganism biology,
were isolated pigmented strains that are resistant to a wide range of the most toxic metals. These are mercury, chromium, copper, cobalt, nickel, and cadmium. Therefore, samples selected on the cliffs of biogeographical polygon are of the considerable interest in terms of isolation of biologically active substances and metal-resistant microorganisms. It is interesting in two aspects. The first is fundamental, how in the most ecologically clean area of the globe in Antarctic appear microorganisms that are resistant to the most toxic metals. And the second appears new prospects to use these microorganisms for creation of new environmental biotechnologies for industrial wastewater treatment against the wide range of metals. Now, step by step, we show the path of profile, sampling and characteristics of position points.
Точка ТСК-2.
Точка ТСК-2.
Abbreviated name of point TSK means T is a point and SK is a rock. In this point we observe black crusted lichens which form a continuous falling film. Passing the cliffs, we see completely naked areas of rocks near the lichens layers of biofilm. These naked areas are formed by spalling of blocks from the cliff surface. And it is also clear that the destruction level of rocks is maximal in biofouling zones.
Это точка ТСК-3. Точка ТСК-3.
Точка ТСК-3.
Точка ТСК-3. Отлично.
The rock is completely covered with black crusted lichens. For comparison, it is the naked rock area of red-brown color. We separated biofilm samples on two parts. One frozen and stored at minus 20 Celsius degree, and another stored in native conditions.
Точка ТСК-4.
Point TSK-4. Here we can see the rock destruction caused by crusted biofouling.
Вот это будет ТСК-5. Вот здесь.
ТСК-5.
This rock is covered with black films of biofouling. The weather quickly and unpredictably changes in Antarctica. The first profile, we pass along the dry rock and bright sunlight. The second profile, we passed on the frosty rock surface and at the horizontal flank snow with rain.
Это точка СТК-7. СТК-7.
Point TSK-7. From top to bottom along the cliff it is shown the patterns of biofouling distribution. At the top on the vertical and negatively inclined profile
are dominated grey and black crusted lichens. And in the bottom of the cliff, on the steeply inclined and vertical surface, there are dense thicket of bushy grey lichens. On this cliff there are areas with 10 or even 20 square meters covered by continuous lichens layer of biofouling. At the first sight, syncrustous lichens, tunnels of fruticose lichens, moss, bright green grass Deschamps Antarctica seems to be delicate and fragile. In fact, they grow firmly into the rock. Fruticose lichens can even be used as hooks in climbing. However, the process of cliff surface colonization is not only limited by the formation of tunnel lichens on the cliff walls. For example, at point TSK8, located at the bottom of the second profile, appears moss formation with square from 10 to 15 centimeters and height from 5 to 6 centimeters. The moss and lichen necromas are accumulated on small shells and in the rock pocket. Further, these necromas transformed into humus, which is a suitable substrate for higher plants, particularly for Deschampsia. In 2011, on the right from the profile 2, we found the bush of Deschampsia in the vertical crack. Thus, at the cliff Scaladrome 2, there are diversified ecosystems represented as crustous and fructicose lichens, mosses, and representative of cereal Deschampsia Antarctica.
ТСК-9.
It is obvious that in the dense biofoulant layers there are concentrated microorganisms and lichens. Microorganisms and lichens produce organic acid, which gradually destroy and dissolve the rock surface. In addition, microbial exometabolites transform the wide range of metal from rocks into a soluble form.
Точка ТСК-10.
Powerful, irrepressible development of ecosystem on rocks is shown in point TSK10. Here, in spite of a negative slope in 20 or even 30 degrees, the rock surface is covered with thick biofoulant layer. Even in crack grow mosses.
Сейчас. Точка Т10. Точка Т10.
The rocks are rapidly destroyed under the influence of organic acids and mechanical breaking of fouling biomass. Now we see the final result of biological attack of rock. Rock layer broke up, exposing the native rocky surface, which also will be colonized by microorganisms, microscopic algae and lichens. Scaladrome 2 is a polygon with position points for sampling and quantitative characteristic and dynamics changes in ecosystem carrying out with a frame of area 1 square meter. The plastic labels with a number of points are fixed on position points of the first and second vertical profiles. In 2011, with the help of square frames with the side of 1 meter, we have received the archive photos of landscape of 12 points from TSK0 to TSK12. Frame scale is equal to 10 centimeters. It allows determining the size and ratio of the surface of naked rock and biofouling formation. Long-term imaging of ecosystem in position points with scaled frame allow us to establish the patterns of formation and distribution of biofouling of cliffs on Scaladrome 2. Antarctic rocky cliffs are ideal objects for studying the influence of complex system of extreme factors on microbial cyanosis. On the cliff Scaladrome 2, the surface is constantly snow-free, so during cold year, microbial cyanosis are available for the influence of UV radiation. Therefore, we can assume that pigmented microorganisms are widely spread on vertical rocks. The pigments protects microorganisms from the harmful effect of UV radiation. Microbial exometabolites mobilize metals from the rocks. Therefore, we can assume the presence of metal-resistant microorganisms in ecosystem of cliffs. Finally, the unequal distribution of organic matter leads to microbial antibiosis as a result of a competition for substrate. It makes easy to isolate the strains of antibiotic producers, antibiotic resistance microorganisms. The experimental data confirm our assumption. Comparison 7 ecosystem, it was established that the pigmented microorganisms dominate in ecosystems of rock cliffs. Comparing the lichen samples from the soil samples, it was shown that the number of pigmented microorganisms on the rocks is several times higher than in the soil. 1 gram of sample taken from the cliff contains hundreds of thousands of cells of red pigmented microorganisms. A number of dark pigmented microorganisms in sample of cliffs were up to 80% from total number of the cells. In this photo, you can see that even in the snow winter, microorganisms and lichens are available for the action of UV radiation. From the diagrams, it is seen that large number of pigmented forms of bacteria, yeast and fungi are present in all 12 points of Scaladrome 2 at 2 profiles. They are colored in black, red, yellow, orange and pink. The height of colored columns is proportional to the quantity of pigmented microorganisms. Thus we can make a conclusion about their widespread distribution on the cliffs of Scaladrome 2. On the next 12 photos is shown the quantity distribution of various pigmented microorganisms on all position points of Scaladrome 2. Of course, the certain relation or a combination of different pigmented forms substantially varies from point to point, but common for all samples was very high content of pigmented microorganisms from some millions to 10 million cells per gram of sample. Representative, or in other words, typical strains of microorganisms were isolated from limiting dilution of samples taken from the cliff. These microorganisms were cultivated in media in the absence of extreme factors. We have isolated the pure yeast cultures, which are the potential producers of biologically active substances, such as melanin, black, and carotene, red. A distinctive feature of our research is a close correlation between the fundamental characteristic of Antarctic microbial ecosystems and practical application of the isolated microorganisms in the novel biotechnologies. Thus, during the study of quantitative relationships of microbial resistance to UV radiation, we isolated a number of microorganisms, superproducers of carotenes, melanins, and possible flavins. Thus, the fundamental research of Antarctic microbial ecosystem are closely linked with the bioprospecting. These are producers of carotenes, melanin, and probably of flavins. From samples selected on cliff, we isolated and identified strain of Exophylla nigra. It is a superproducer of biologically active substances, melanin. We have studied the physical-chemical properties of black pigments that are synthesized by yeast isolated from the cliff Scaladrome 2. Further, we will show that this pigment is melanin. According to the absorption spectrum, its pigments are identical to the melanin that produced by Biotechnological Industry. Industrial preparation of melanin were obtained from Professor Tatyana Beregovaya. Antarctic yeast are super producers of melanin. the melanin wilt in the range from 6 to 10% of absolutely dry yeast biomass. During the lysis of yeast cells, the melanin accumulates in the culture liquid in the form of large aggregates. We compared the properties of melanin isolated from Antarctic black yeast with melanin isolated from the typical well-known producers. As it is shown from the table, the properties of the both melanins are completely identical. Also, we isolate pink metallotroph bacteria, producers of carotenes. Carotene purified preparation of metallotrophic bacteria has a characteristic typical absorption spectrum. Carotene yield is from 0.23 to 0.35 milligrams per 1 gram of absolutely dry biomass of cells. Yeast containing beta-carotene of bright red color are widely spread on cliff. At some points on Scaladrome 2, the number of red yeast in the range from some millions to 10 million cells per 1 gram of sample. Carotenes have high antioxidant activity, which is manifested at the activation of highly reactive free oxygen radicals. Therefore, red yeast isolated from cliff of Scaladrome 2 can be considered as a potential producers of beta-carotene for industrial biotechnologies. Non-pigmented metallotrophic bacteria from cliffs were less resistant to UV radiation. For them, parameter lethal dose 99.99, that is the death of 99.99% of the cells, were determined with a UV dose of 80 joules per square meter. Pigmented strains of metallotrophic bacteria were 3 times more resistant to UV compared with non-pigmented ones. The pigmented forms LD 99.99 was established at a dose about 250 joules per square meter. For black melanin containing yeast isolated from Scaladrome 2, the parameter LD 99.99 was at a dose of UV from 700 to 800 joules per square meter. Red yeast carotene containing yeast shows the maximum resistance to UV radiation. For them, the parameter LD 99.99 is defined at UV dose about 1500 joules per square meter. Black yeasts are resistant both to UV radiation and toxic metals. For them, the maximum permissible concentration of chromium-6 was up to 7500 part per million, of nickel cation up to 250 ppm and up to 1000 ppm of cobalt cation. It should be noted that given metal concentration are in 2 orders higher than bactericidal concentration, lethal for the most chemoorganotrophic microorganisms. For strain number 36, the increase in the concentration of cobalt in nutrient medium leads to stimulating of melanin synthesis. It is known that melanins are an effective sorbents of metals. Therefore, Antarctic melanin containing microorganisms resistant to metals can be used to develop new environmental biotechnologies for industrial wastewater treatment from toxic metals. Melanin isolated from Antarctic yeast has another important practical applications. Professor Tatiana Beregovaya demonstrated that Antarctic melanin prevents the development of ulcers, erosions and hemorrhages in the stomach mucosa of rats, precancerous state. Typical microorganisms of cliff Scaladrome also shows a high resistance to 5 the most toxic metals. These are cations of mercury, copper, cobalt, nickel, and anion of chromium-6, chromate. These metals are metal oxidizers, metal substitutors, and metals of combined action, being both metal oxidizers and metal substitutors. Testing of 10 typical Antarctic strains shows a high level of metal resistance. For mercury, maximum permissible concentration was 50 ppm, for cobalt was 100, for nickel and copper more than 1000 ppm, and for chromate anion in recalculation on concentration of chromium-6 was even 10,000 ppm. As we noted, on the vertical cliffs there is an equal distribution of organic compounds, carbon and energy source for microorganisms. This leads to the strong competition of microorganisms for power supply and therefore to antibiosis formation. Antibiosis has two aspects, that is, synthesis of antibiotics and antibiotic resistance. As example, the isolated by Olga Gosoflyak and Elena Kiprianova Antarctic strain Pseudomonas putida shows a high level of antibiotic activity in relation to 4 test cultures. It should be emphasized that at temperature of 26 Celsius degree, level of antibiotic activity was minimal. However, the decrease in cultivation temperature up to 4 Celsius degree leads to increase of antibiotic activity. Growing strains at so-called Antarctic temperature cast increasing diameter of growth inhibition zones of test culture, which made from 10 to 20 millimeters. As expected, the typical microorganisms showed high resistance to a wide range of antibiotics. We have tested the resistance of 9 typical strains of Scaladrome 2 to 22 antibiotics of broad spectrum. The height of columns on diagram shows the resistance to 9 strains to each antibiotic. From this data follows that the typical microorganisms exhibit high resistance to a wide range of antibiotics. Only 2 antibiotics, gentamicin and ciprofloxacin, inhibited the growth of 9 typical cultures. Further, on the next 4 photos, we show the examples of microbial resistance and sensitivity of microorganisms to antibiotics. The diameter of sterile zones around the disc with antibiotic is proportional to the inhibitory effect of antibiotics. Thus we show the polyresistance of microorganisms on Antarctic cliffs on Scaladrome to UV radiation, the most toxic metals, and wide range of antibiotics and also high levels of antibiotic activity. The obtained data from fundamental characteristics, structure and functioning of microbial ecosystem can be effectively used in applied practical microbiology. The effectiveness of bioprospecting is determined by the comprehensive of fundamental researches. For example, in the study of UV resistance was carried screening of pigmented microorganisms and the development, the collection of these microorganisms. The next step is a comprehensive study of pigmented microorganism properties. And as a final result of our researches is determination of various range of Antarctic strains in industrial biotechnologies. So, on the base only one of melanin producer, Exophiala nigra, it is possible to create a sunscreen cosmetic creams, granule preparation of wastewater treatment from toxic metals, new anti-ulcer and anti-cancer medications. There is nothing more practical than a good theory. Another example of the continuous connection of fundamental and applied researches. As studying the resistance of typical Antarctic microorganisms to toxic metals, has been shown that the culture Enterobacter hormechii and Bryobacterium antarcticum are resistant to high concentration of mercury, copper cations and anion of chromium-6. Thermodynamic calculations show that at oxidation of organic compounds, the microorganisms can reduce soluble copper-2 cations to insoluble copper-1 compounds.
We have shown that the Biobacterium antarcticum, in a direct flow compact pilot plant with length of 1 meter, reduced copper cation concentration in water from 325 to 0.1 ppm. This process takes place due to the reduction of copper-2 to insoluble copper-1 oxide. For cell immobilization, the substrate is potato waste. At the same time, this substrate is carbon and energy source for bacteria. This technology provides to obtain 4 positive effects. The first is the destruction of the environmentally hazardous solid waste, i.e. waste potatoes. The second is withdrawing of toxic metals from the wastewater. The third is obtaining valuable concentrate of copper, and the last obtaining of purified water available for closed technological cycle. On the ground of database obtained from the complex study of structure and functioning of Antarctic microbial ecosystem, we formulated strategic perspective of fundamental and applied researches in Antarctic. Fundamental and applied researches we carried out on 3 levels. The first is microorganisms, the second is phenomenon, the third is processes. On fundamental level, microorganisms, we study the ecological, trophic, physiological and other groups of microorganisms, but also we create the collection of typical microorganisms. Phenomena. We study the homeostasis of microbial ecosystems under the influence of the complex of extreme factors, such as UV radiation, antibiotics and toxic metals. Processes. We study the role of the microbial biota in the global biogeochemical and climate processes. Applied researches include microorganisms. We develop the collection of Antarctic microorganisms, biologically active substances producers. Phenomena. That is development of novel environment protection microbial technologies and biotechnologies of biologically active substances production. Finally, processes. Creation of predicting models of the biogeochemical and climate processes in Antarctica. The leading Ukrainian microbiologist, director of the Bolotny Institute of Microbiology and Virology of National Academy of Sciences of Ukraine, Professor Academician Valentin Podgorsky, estimates 10 years microbiological researches in Antarctic.
The researches of Antarctic microorganisms that were carried out at the Institute of Microbiology and Virology are related with 2 directions. On one hand, the studying of their habitat, and on the other hand, their role in nature, metabolic processes and physiological features. I mean, the fundamental researches open up new concepts, new cognitive processes in microbial vital activity. A very interesting fact that a lot of bacteria and yeasts isolated from Antarctica are resistant to high concentrations of various toxic metals. It is also important that these microorganisms are able to synthesize a variety of biologically active substances. First of all, it is their properties to synthesize different pigments. These pigments are antioxidants, help to improve the vital activity of organisms, of higher animals and human beings. I think such research is carried out at the Institute of Microbiology and Virology of the National Academy of Sciences of Ukraine under the leading Doctor of Technical Sciences Alexander Tashyrev will contribute to further understanding of structure and function of microbial ecosystems, allow to create new biotechnological processes on the basis of their vital activity.
чтоб не было следов. Повсюду подмели, ругайте же меня, позорьте и трезвоньте мой финиш горизонт. А лента край земли, я должен первым быть на горизонте. Условия пари одобрили не все, и руки разбивали неохотно. Условие таково, чтоб ехать по шоссе, и только по шоссе бесповоротно. Наматывая мили на кардан, я еду параллельно проводам. Я прибавляю газ до иступления в местах, где ограничено движение. Я знаю, мне не раз в колеса палки ткнут, догадываюсь, в чем и как меня обманут. Я знаю, где мой бег с ухмылкой пресекут и где через дорогу трос натянут. Но стрелки я топлю на этих скоростях, песчинка обретает силу пули, и я сжимаю руль до судорог в кистях, успеть, пока болты не затянули. Наматывая мили на кардан, я еду вертикально проводам. Завинчивают гайки побыстрее, не то поднимут трос как раз где шея. И плавится асфальт, протекторы кипят, под ложечкой сосет от близости развязки. Я голой грудью рву натянутый канат. Я жив, снимите траурные маски. Кто вынудил меня на жесткое пари? Нечистоплотный в споре расчетах. Азарт меня пьянит, но я твержу, смотри, и тормози на скользких поворотах. Наматывая мили на кардан, назло канатом, тросом, проводам, вы только проигравших в горизонте, когда я появлюсь на горизонте. Мой финиш горизонт по-прежнему далек. Я ленту не порвал, но я покончил с тросом. Да трос не пересек мой шейный позвонок, но из кустов стреляют по колесам. Меня ведь не рубли на гонку завели, меня просили, ник не проворонь ты. Узнай, а есть предел там на краю земли, и можно ли раздвинуть горизонты? Наматываю мили на кардан, и пулю в скат влепить себе не дам. Но тормоза отказывают схода, я горизонт промакиваю хода.
А ну-ка песню нам пропой, веселый ветер, веселый ветер, веселый ветер. Моря и горы ты обшарил все на свете, и все на свете песни слыхал. Спой нам, ветер, про дикие горы, про глубокие тайны морей, про птичьи разговоры, про синие просторы, про смелых и больших людей. Кто привык за победу бороться, с нами вместе пускай запоет. Кто весело смеется, кто хочет, он добьется, кто ищет, он всегда найдет.
We are looking for the international collaboration in the field of comprehensive research for extremal microbial ecosystems. Welcome.